Hi, my name's Chris Martin and I'm a neuroscientist working at the University of Oxford. I've been lucky enough to be invited to this year's Lindau Nobel meeting, and I can't wait to meet all the other young researchers and of course the Nobel laureates themselves. My research focuses on functional brain imaging, and in particular I want to find out more about what brain imaging methods can tell us about how the brain works under normal conditions, but also what goes wrong in diseases of the brain. The general idea behind functional brain imaging is that as a particular part of the brain becomes more active during a task, or perhaps after a drug has been administered, then increased blood flows to that region, just as you exercise a muscle in your body, so in your arm, increased blood flows to that particular muscle. So by using methods that detect changes in blood flow, which is what fMRI, for example, does, we can detect changes in brain activity that we can ascribe to either demands of the task or, for example, the effects of the drug that's been administered. Now, one of the limitations of this method is that we don't know very much about how the changes in blood flow are actually driven by changes in the activity of the brain's cells, that is, the neurons. So I use a range of different recording methods to look at both the activity of the cells themselves in the brain, but also changes in blood flow, and hopefully by doing that I can learn a lot more about the relationships between brain imaging signals and the actual activity of brain cells.